Next, we're going to hear from Dr. Subhrangsu Mandal. He's presently a professor at the Department of Chemistry and Biochemistry at the University of Texas at Arlington. Today, he's speaking on unfolding DES epigenome, impacts on chromatin genes and non-coding RNA. All yours, Subhrangsu.
Hi. Thank you, Shu. And can you hear me? Can you hear me?
Yes.
And the slides are visible?
Yes, you could put it full screen by clicking on slideshow.
Okay, and so first of all, I'd like to thank the organizer. I think this has been really an excellent and educational day for me from the morning till now, and a lot of interesting stuff on endocrine disruption and DES, transgenerational and multi-generational studies. So my lab, primarily, I'm a biochemist and I'm relatively new, I would say, in the field of endocrine disruption and DES epigenetics. But my lab primarily studies the chromatin modification, histone modification, gene regulation in human and epigenetics mechanism. And we have spent a good amount of time studying a class of histone methyltransferase, which are called mixed lineage leukemia. Of course, they are mutated, associated with leukemia. And lately, my lab has been extending our studies, unfolding the function of non-coding RNA, which I'll talk a little bit today. And I got into this endocrine signaling, endocrine disruption, because we find that some of the critical histone modifying enzyme, methyltransferase, like mixed lineage leukemia, which was our major interest, and they appear to interact with nuclear hormone receptor and control the hormone-dependent gene expression. So in my lab, we have showed that these histone methyltransferase actually interact and coordinate with estrogen receptors and control the estrogen-dependent gene activations involved in cholesterol metabolism, neuroprotection. And with that, because we are studying the estrogen, we also started looking the epigenetic mechanism of endocrine disruption by some of the endocrine, estrogenic endocrine disrupting chemicals. And I'm sure for this audience, it does not need an introduction, but I think today there are people from maybe outside. So what I want to briefly summarize is that the genomic material, the DNA, which contains the genetic information and so on, they're actually packaged inside the nucleus of our cell with variety of proteins, which are called histone proteins. And for a long time, people knew that these histone proteins, primary function of the histone proteins is to compact the large piece of DNA in the tiny nucleus. But now we know that these histone proteins actually goes through different type of modification. They have much more dynamic function beyond just the packaging large piece of DNA. So amino acids in histone proteins goes through methylation, acetylation, phosphorylation, and different types of, these are called post-translational modification. And along with the genetic information, these post-translational modification contribute how the gene will be turned on and turned off. So this histone modification plays a critical role in terms of gene activation, gene silencing. But last about 10, 15 years of research shows that along with this histone modification, we have these new class of transcripts which are called non-coding RNA, which are integral component of this gene regulation machinery. So I want to draw your attention a little bit on some of the histone modification. We have been working on several, but today I'll talk one of the histone methylation, which is histone H3 lysine 27 methylation. And this methylation is critically linked to gene silencing. We have heard a lot of talks on CpG and methylation and so on, which are linked to gene silencing and controlling the genomic epigenomic activity. Along with the CpG and methylation, the histone H3 lysine 27 methylation is critical for controlling gene silencing, and of course the demethylation will lead to gene activation. And this methylation is introduced by an enzyme is called EGH2. This is an enzyme, it's an histone methyltransferase which methylates histone H3 lysine 4 and controls the histone methylation, and that's how it induces gene silencing. It has close collaboration with DNA methylation and others. And this is, I should emphasize that this methylation is very, very critical in the early stage of development. And the malfunction or overexpression of this histone methyltransferase EGH2 is closely linked with variety of cancer, including breast cancer, ovarian cancer, and so on. In addition to this histone methyltransferase EGH2, I want to introduce a non-coding RNA which is called HOTAIR. Okay, I don't know whether I have time to introduce a lot about non-coding RNA, but non-coding RNA is a class of transcripts which are coded by the genome, but they are not mostly translated into protein. For a long time, people thought they have no function. They are called junk DNA, junk transcript or noise in the cells. But now we know that these non-coding RNA, even if they are not translated into protein, they have critical function in controlling the chromosomal functions. And their malfunction will lead to different types of abnormal gene regulation and disease. And it is very important to emphasize that this non-coding RNA, HOTAIR, actually physically interacts with this histone methyltransferase EGH2, and they together control the histone methylation and gene silencing. So again, like EGH2, the HOTAIR expression is highly overexpressed in variety of cancer. So because we are studying the epigenetic regulation of gene expression, and HOTAIR appear to be involved in epigenetic histone methylation and gene silencing, so we started looking at the function and regulation of HOTAIR. We have published several studies on the HOTAIR. This is non-coding, and we show that it is overexpressed in breast cancer. It is regulated by estradiol. It controls the histone H3 lysine 27 methylation in the chromosome. But what I want to show, because this is regulated by estradiol and it is essential for cancer. Okay, it's a tumor suppressor basically. It's not a tumor suppressor, it's an oncogene, but it suppresses the tumor suppressor expression. That's how it contributes to cancer. So because it is oncogenic and controlled by estradiol and overexpressed in variety of cancer, we thought, let's see whether this particular oncogenic non-coding RNA is controlled by the estrogenic endocrine disrupting chemical like BPA and DES. So we have done all the cell culture, and this data is actually animal studies where we have exposed the ovarectomized rats with either BPA or DES or estrogen independently or in combination. And what we see, the HOTAIR, the non-coding RNA, its expression is elevated in the mammary tissue by estradiol, and the elevation, the HOTAIR RNA is also elevated by the DES even in the absence of estrogen. So think about this. This is an oncogenic RNA. That means its overexpression would facilitate the tumor growth. And now, upon exposure to DES, we see that this non-coding RNA expression is elevated. So that kind of suggests that it may have a potential role in inducing the tumor growth and so on. Another piece of studies in the same direction, what we show that EGH2, which is a histone methyltransferase, is a very critical player in cancer as well as early stage of development, and we see that, and it interacts with HOTAIR, we see that EGH2 expression is also elevated in the mouse, the rat mammary tissue upon exposure to estradiol, which is kind of expected, but what we saw is the elevated level of expression of EGH2 in the mammary tissue, both by BPA and DES, in the absence of estrogen. So what you see, the staining, this is a control tissue, this is with estradiol, the level of overexpression, and this is by BPA and DES. So this kind of suggests that even when the actual hormone is not there, when we are exposed to some of these estrogenic endocrine disrupting chemical like DES, BPA, it can also induce the expression of some of the genes which may contribute to the cancer and developmental defect. So I did not want to go too much detail of our study, but we also looked at expression of the developmental genes, especially the Hox genes, and along with the HOTAIR EGH2, we see that many Hox genes are also affected by BPA and DES. So at this stage, we are more on the looking at the single gene expression level, but lately, because at this point we started to understand that how bad DES or BPA exposure or endocrine disruptors may be affecting our health, development and health. So we started looking at the genome-wide level, the impact of some of these endocrine disrupting chemicals. So some of these studies at this point we are in progress, some of the data we have, but more experiments are going on. So what my lab is right now trying to do is that we try to understand what are the coding as well as non-coding RNAs, non-coding genes expression may be affected by exposure to DES or BPA in vitro, that means in cell lines, but also in vivo in the mouse model and rat model. So basically what we are going to do, what we are doing is right now exposing the cells with these drugs, isolating the total transcriptome and doing the RNA sequencing and finding the coding as well as non-coding RNA expression. And once we have these coding and non-coding RNA in hand, we actually have done a couple of RNA sequencing analysis right now, and we are actually classifying them based on signaling pathway. Basically, my goal is to see upon exposure to DES, what specific cell signaling pathway may be affected. And based on that, we can actually try to see how they contribute to the cancer or developmental defects or some of the risk factors. Also, I realize that finding a biomarker, okay, critical biomarker for DES exposure or BP exposure is very critical in understanding the risk factor or assessing the risk factor or more information. So we are actually heavily focused on, based on these non-coding RNA or coding RNA, we want to actually develop the blood-based or urine-based biomarker for DES and BP exposure using both animal model and cell culture. Eventually, actually at this point, this is a project which is in progress, where we are actually moving to the actual DES exposed population. I want to actually share one point which I learned last couple of months and less than few years, that although we try to do a lot of experiment in mice, and I see that we do like to trust that mice phenotypes, functions are can be extended to human. But I can tell that based on my experience, and I think many of you will agree, that long non-coding RNA, which is about 70–80% of the transcriptome, what we have in the cell or in our physiology, they are not conjured in mouse to human. So even if we identify a lot of non-coding RNAs which are responsive to DES or a lot of phenotypes controlling phenotypes. Once we try to extend the studies to human, I could not do that. I don't find most of the long non-coding RNA especially is not conjugate. So at this point, we are completely switching the genome-wide profiling to the DES-exposed and control population and DES-exposed population, both male and female, and we want to focus on multi-generational studies. So basically, we want to isolate the blood samples and do the non-coding RNA profiling and look for potential biomarkers. Yes, again, with the hope that we can guide the therapeutics in future. Also, I have some collaboration with some of the physiologists, and where we actually want to, the patients who are especially Dallas-based, I'm in the Dallas-based area, so we want to recruit whoever agrees. We want to study some of the physiological parameters, especially look at their neurocardiovascular function, immune functions, and bone muscle functions.
Dr. Mandal, I'm so sorry. We're over time, so I'm going to ask you just to wrap up in about 10 seconds, and then we have to move on. I'm very sorry.
This is the last slide. So this is where exactly we are heading, and hopefully in a couple of months to years, we have more to talk. Thank you. And
Thank you.
these are the acknowledgements. Thank you for the patience.
Thank you. That was actually is very interesting, and the biomarker is particularly interesting. So good luck.